Okay, we'll go ahead and continue on with Chapter 5 here and finish that up. Now, in the fermentation process, after glucose is broken down into pyruvic acid will then undergo fermentation. We know that process of breaking down glucose is glycolysis. Fermentation does not require oxygen, doesn't require electron transport chain, as we've talked about earlier with the aerobic process and the Krebs cycle, as we alluded to earlier. It uses organic molecules as the final acceptor. In fermentation, pyruvic acid accepts electrons, hydrogen, and it also is turned into various end products such as lactic acid or lactic acid and ethanol, and alcohol. And again, those are the two processes we talked about before: homolactic fermentation, heterolactic fermentation, one of which produces lactic acid only, and the latter produces acid alcohols as well as lactic acid. Fermentation releases energy from oxidation of organic molecules, does not require oxygen. Big difference between it and the Krebs cycle. Does not use the Krebs cycle or the electron transport chain, as we've talked about earlier. It uses organic molecules as the final acceptor. The process of fermentation and the various microorganisms that utilize it, the ones we'll be talking about mostly as we get later on into the lectures, especially related to disease processes: Streptococcus, Bacillus, we've referred to already, propionic bacterium, Clostridium. Clostridium causes a significant number of diseases, and we'll talk about that in more detail. Escherichia coli, Salmonella, Enterobacter are common organisms that utilize pathway of fermentation. Fermentation end products: lactic acid, we may be familiar with ethanol, CO2, propionic acid, butyric acid, ethanol, and lactic acid are different end products that we see in this fermentation process. And alcohol fermentation produces ethanol alcohol or ethyl alcohol plus carbon dioxide. And again, we've already talked about lactic acid fermentation and the products there: lactic acid, again, lactic acid and other compounds such as alcohol. But this is just an illustration, again, out of your book, showing you the steps of the process of the fermentation pathway. Again, I am not going to hold you to task in knowing each of the steps of the fermentation process. I would expect you to recognize that this process takes place in the absence of oxygen, and why it's important, because it gives an alternative pathway for other organisms to be able to produce energy. Some microorganisms basically can produce energy from the breakdown of fats. So we'll have to mention lipid catabolism a little bit. Again, you can get carbon compounds from other sources. Doesn't have to be glucose. Doesn't have to be a 6-carbon sugar. It can be from lipids, which are long chains of fatty acids. It can be from proteins as well. The breakdown, the metabolism of these various organic compounds is a little bit different, but microorganisms can utilize them as well. Some microorganisms produce extracellular enzymes, lipases, ASE, again, lipases, which basically break fats into fatty acids and glycerol. The components are then metabolized separately. So fatty acids are oxidized basically by a process referred to as beta oxidation. You need to know that fatty acids oxidized by beta oxidation, in which carbon fragments are basically removed 2 at a time to form acetyl coenzyme A. You've heard that term before because it's utilized in the Krebs cycle. And so those molecules then enter into the Krebs cycle. Again, instead of using carbohydrates, instead of using glucose, we're now using lipids. Why? Because there's an enzyme, lipases, that help produce extracellular enzymes, these extracellular enzymes that help to break down fats. Glycerols form one of the intermediates for glycolysis, and then it's further oxidized. So in metabolism or catabolism, when we talk about energy sources, not only for the biological system, we can use lipids, we can use carbohydrates, and a little bit we'll talk about the use of proteins as well. But lipids, basically broken down by lipases. Lipases generate glycerol fatty acids. And look at what both glycerol and fatty acids can do. They too can go through a series of enzymatic steps. Fatty acids go through beta oxidation, enter into acetyl-CoA, go through the Krebs cycle, boom, produce ATP on the other end. Glycerol, also a product of lipase. The reaction that takes place goes through, again, a series of enzymatic steps. You're familiar with glycolysis already. Pyruvic acid, acetyl-CoA goes through the Krebs cycle inside the, whether it's the microorganism or whether it's inside the human system, and again produces energy on the other side in the form of ATP. Now, digress here for a second. Why does certain types of diets work that you do not utilize carbohydrates and you use fats instead? Because we can use in cellular metabolism, if we have to use lipids. Lipid is not the first choice of an energy source in the human system. The first choice is carbohydrates. And so if we use carbohydrates, fats will continue to be stored. But if we only go to fats and we start using fat catabolism, then we can obviously get energy from the utilizations of fats as well, and carbohydrates will be spared and not used, and the body will by deference produce these substances using lipid catabolism. Now, I also had alluded to earlier about protein catabolism. Again, primary source of energy, you want that to be carbohydrates, but if carbohydrates are missing in the diet, we can alternatively use lipids. We have lipids stored, and so the body will use our stored lipids as well for an energy source, or we can use protein catabolism. Now, in the human system, protein is one of the substances that, organic substances, that the body will use as a last resort. Typically, the body doesn't want to use protein for an energy source, but it will in stressful situations or when there is an absence of carbohydrates and an absence of lipids. There are proteases and peptidases. Now, remember we talked about lipases for the lipids. We're talking about proteases and peptidases, basically that are extracellular enzymes produced by some microorganisms, microbes, to break down proteins which are too large for these microbes to use unaltered into components of amino acids. Amino acids are first deaminated, another term you need to be familiar with, deaminated, which means ammonia groups will be removed. They are also decarboxylated, another term you need to know because the functional group is removed, and then they will enter the Krebs cycle in various ways. Proteins, extracellular proteases, again, break down to amino acids. We can't use the amino groups, so then they have to be deaminated, amino groups taken away. Deamination, decarboxylation, dehydrogenation has to take place. Organic acids are produced, enters into the Krebs cycle. So now we have both carbohydrate catabolism, we have lipid catabolism, and we have protein catabolism, all of which can be used for an energy source. Now, some microorganisms, because we now know they require specific energy sources, some specifically require or can only use carbohydrates, some specifically require or only use proteins, some specifically require the use of lipids. They also will produce byproducts. In testing, if we're trying to identify particular microorganisms, then now what we're able to do is use specific biochemical tests to be able to identify microorganisms based upon what their sources of energy are or based upon what their end products may be. So not only can we inhibit the growth of certain microorganisms by limiting their energy source or by somehow breaking up those enzymatic steps that are required for these microbes to be able to survive, but now we can also identify these same organisms based upon what they generate and what types of reactions are required for them to be able to carry out their life. Again, they can ferment glucose. We can answer the question yes or no. Can they utilize the citric acid cycle as a sole source of carbon? Then it helps us to know this is a particular microorganism such as Shigella, or is it a particular organism such as Salmonella, because it produces various byproducts. Same process can take place when we look at does the organism utilize the citric acid cycle? Yes or no? Yes, then we know we can defer for organisms that produce a different byproduct, and we look at two organisms at the end of the chain, Citrobacter and Enterobacter. if they do not use the sole citric acid cycle as their sole carbon source, then we think of organisms such as Escherichia coli. So again, now I'm not showing you this illustration for you to sit there and try to memorize those steps. I'm showing this so you can take the general concept of we can identify organisms based upon their energy source and based upon the byproducts that are produced at the end of this cycle. And it helps us not only in identifying the organism that we're talking about, but it also helps us when we want to manage bacterial growth or microorganism growth. It helps us to be able to know what is the energy source that's required for these various organisms. Photosynthesis, another process that you may be familiar with if you've taken previous biology courses. Again, now we've discussed some alternative pathways, again, aerobically, anaerobically, looking at the Krebs cycle, looking at fermentation or glycolysis in the Krebs cycle. Now, this energy source is a result of photosynthesis, again, which electrons are basically taken from hydrogen atoms of water and incorporated into sugar. Formation of ATP in this manner is called photophosphorylation, but requires light as the conversion. Conversion of light energy into chemical energy, ATP, and then light-dependent light reactions, and then synthesis, basically where you're fixing carbon into organic molecules. Light-independent dark reactions also has a particular cycle, a series, again, of sequences, reactions that take place, known as the Calvin-Benson cycle. And then these can be done in the presence of oxygen or in the absence of oxygen based upon the source. Cyclic phosphorylation, basically electrons return to chlorophyll. This is an example where you can see the electron transport chain in a microorganism that would involve chlorophyll. Again, we see that the electron carrier is still utilized. The end product is still going to be ATP. But light basically now excites the electrons, utilizes the electron transport chain, and energy produced in the form of ATP. But you see this type of process, cyclic phosphorylation, would require the use of the light energy as the source, and then again a different type of electron carrier. In non-cyclic photophosphorylation, electrons are basically incorporated into NADPH. NADH you've seen is a high energy compound. Now we're phosphorylating it, producing NADPH. Again, light is required. You see chlorophyll is still used. The electrons are basically excited, still pass down the electron transport chain. NADPH is given off, and ATP energy production occurs in non-cyclic photophosphorylation as well. But again, light is required, chlorophyll is required for this pathway to be able to work. And then the concept again of chemiosmotic proton pumps we already talked about, but again in these same types of organisms and even in organisms not chlorophyll to generate electrons through this process, again where the energy that's produced is by the charges that will accumulate on various sides of the membrane. And that's just a particular organism that's used to generate that. Hello.